Hi, this is Tutor Ivan Serunjoji, and today in this tutorial we are looking at how to describe the biochemistry of C4 plants / the H/SLAC pathway. How to describe the biochemistry of C4 plants / H/SLAC pathway. Description of the H/SLAC pathway, an overview. So by the end of this tutorial, we shall have covered these questions and we shall have settled them. The first question is where is carbon dioxide fixed initially? As we looked at in our previous tutorial, the C4 plant anatomy is made up of dimorphic chloroplasts. So we want to see where is the initial fixation of carbon dioxide occurring. That is our first question. Our second question shall be, how does photosynthesis occur in dimorphic chloroplasts? How does photosynthesis occur in the dimorphic chloroplasts? We want to look at this magic. Then another one is, how does the C4 pathway differ from the C3 pathway? Basically, a comparison shall be made after us looking at this pathway and analyze it with the first pathway we made or we looked at, that is the C3 pathway. Let's go. Before we look at the real biochemistry of C4 plants, we need to first look at the arrangement of cells in the C4 leaf. So as you look at this pictorial, we are looking at a leaf arrangement where the outer ring is made up of what we call the mesophyll cells, and these mesophyll cells have PEP carboxylase. As we shall see, PEP is phosphoenol pyruvate carboxylase. This is for the reaction of carbon dioxide and PEP, that is phosphoenol pyruvate, to form for us a 4-carbon molecule that we shall look at in our next slide. Then this is covering another inner ring of what we call the bundle sheath cells. Now in these bundle sheath cells, we have what we call rubisco, that is ribulose bisphosphate carboxylase oxygenase. So this is an enzyme, a protein in nature with 2 active sites, and the active site which is very active here is what we call the carboxylase part of it, that is the active site that fixes carbon dioxide. For the reaction of RuBUP, that is ribulose bisphosphate with carbon dioxide, released from the C4 compound, that is the carbon 4 compound formed previously in our mesophyll cells. Then, still looking at the leaf anatomy of C4 plants, the close association of these 2 dimorphic chloroplasts found in these different cells, we see that they are having a close association. They are close together. This permits carbon dioxide pumping from the mesophyll cells to bundle sheath cells for the Calvin cycle to occur. So we know that the rubisco is an enzyme that runs the Calvin cycle the way it is in C3 plants. Now, this close association of the bundle sheath cells and the mesophyll cells allows for a mechanism, what we call the shunting of carbon dioxide released from the mesophyll carbon 4 molecule formed to the bundle sheath cells, as we shall proceed and see in the biochemistry. Let's go. description of the Arch-Slack pathway. Description of the Arch-Slack pathway. In other words, the biochemistry of C4 photosynthesis. Let's see what really happens. Initially, carbon dioxide diffuses into the mesophyll cell. Now, in the mesophyll cell, carbon dioxide is fixed and accepted by a molecule called PEP. A molecule called PEP, that is phosphoenolpyruvate, under the influence of an enzyme called PEP carboxylase. In other words, PEPCO. That is PEP, all capital letters, CO, PEPCO, PEP carboxylase. Now, this enzyme catalyzes the fixation of carbon dioxide with PEP, a 3-carbon compound, to form for us oxalacetic acid or oxaloacetate, a 4-carbon compound. Now, this is the first stable compound formed in C4 plants, hence the name carbon-4 plants, oxaloacetate. In short, OAA, OAA, O coming from O here, then acetate, that first A, then the A on acetate. In other words, if it is oxalacetic acid, so we have the first O making it, then the first A on acetate, the first A on acetic, then the first A of acid. Now, what happens? Oxalacetic acid is then reduced to malic acid or malate, still a 4-carbon compound. So, oxalacetic acid is reduced to malate, a 4-carbon compound, using hydrogen from NADPH, formed during photophosphorylation in the light-dependent stage of photosynthesis. Here it is not indicated. So, we find that oxaloacetate is reduced to malate, a 4-carbon compound, using hydrogen from the light-dependent stage, that is the product known as NADPH. What really happens next? Remember from the cross association, we saw that the carbon which was initially fixed with the PEP, that has led to the formation of oxalacetic acid, oxaloacetate, a product that has been reduced to form malate, has to be shunted or exported to another special type of cells with chloroplasts known as the bundle sheath cells, these ones. So, malate is then shunted or exported to the bundle sheath cells, is the chloroplast found in the bundle sheath cells, where it is decarboxylated, removal of carbon dioxide. So we are seeing carbon dioxide being released in the bundle sheath cells from malate. After the process of decarboxylation, we yield pyruvate, a 3-carbon compound. Remember, we have split the 4-carbon compound by removing 1 carbon with oxygen, that is forming a molecule of carbon dioxide, leaving behind the 3-carbon compound known as pyruvate. The resulting carbon dioxide is then entered or enters the normal Calvin cycle. Now, this is the Calvin cycle which occurs normally in C3 plants. We are seeing it in C4 plants occurring in another type of cells known as the bundle sheath cells, or in the bundle sheath chloroplasts. Now, in this Calvin cycle and in this bundle sheath chloroplasts, we find another enzyme known as lobulose bisphosphate carboxylase. This is the enzyme which catalyzes the fixation of carbon dioxide to form for us the sugar after the Calvin cycle. So you know that in the Calvin cycle, carbon dioxide is fixed to rubop, that is rubidose bisphosphate, forming TG, triglycerate, among other products, until we form the carbohydrate or the sugar. Now, the sugar formed is then translocated through the vesicular bundles, basically in the phloem cells, to places where it is needed for storage, maybe in the roots, in the leaves, it stays there and is converted into starch, among others, or in the respiring cells of the plant. Now, what really happens to the pyruvate formed from the decarboxylation of malate? Pyruvate diffuses back to the mesophyll chloroplasts, where it is used to regenerate the molecule, the starting molecule, that is a 3-carbon compound known as phosphoenolpyruvate, PEP. Now, that regeneration requires ATP energy from the light-dependent stage of photosynthesis, where it is hydrolyzed to form PEP, and hence the cycle continues. We should note that the Calvin cycle also utilizes ATP energy, and therefore carbon dioxide in C4 photosynthesis is fixed twice. We saw it being fixed with PEP in the mesophyll chloroplasts, and then during the Calvin cycle it has to again be fixed with rubop. And this requires energy, which is twice that found in C3 plants. Remember, in C3 plants we only have the Calvin cycle. Another thing we need to note is PEP is very efficient at fixing carbon dioxide, and therefore C4 plants have higher energy. They have a higher energy as compared to C3 plants, that is wheat, plants like maize, and all that. They have higher proportions of carbohydrates, that is energy. They store a lot of food, and therefore that's why wheat products are very common. We use wheat powder or flour to make chapati. Then for maize, we also have posho from maize, which is a very, very common food. In our next episode, we shall identify the differences between C3 and C4 photosynthesis or plants. There we shall digest deep into the enzymes found in these plants, and we shall see their significance. Thank you for watching. Don't forget to subscribe by clicking the subscribe button in order not to miss any of our tutorials at Easy Science International. May the Almighty God bless you.